If you watched part 1, you already know where the bioinformatics jobs are, job types, and what they pay. And once they see those job posts, they hit apply button. You see candidates applying to 50 or 80, even 100 jobs, and hearing nothing back. Not because they're not smart, but because their skills look generic. Their GitHub looks unfinished, and their resume blends in with everyone else. In this video, I'm going to show you what actually makes recruiters stop scrolling, the specific programming skills that matter, the 3 projects your portfolio must have, the resume formula that makes you stand out instantly, and how to position yourself so opportunities come to you, not the other way around. By the end of this video, you'll have a clear 4-week action plan to become hire ready. Let's get into it. All right, let's talk programming. Everyone says learn Python, but that's like saying learn English. It's way too broad and doesn't help you actually know what to focus on. Here's the reality. 75% of bioinformatics job postings mention Python. 60% mention R. 45% mention Bash. So yes, these languages matter, but here's what you actually need to know. For Python, not web development or game development Python, you need bioinformatics Python. You need to handle real biological data, parsing FASTQ or VCF files, writing reusable scripts, automating full RNAseq workflows, and optimizing large datasets so your code doesn't crash. R is huge for statistical analysis. If you're analyzing RNA-seq data, you're going to use bioconductor packages. Correct DESEQ2 contrasts, batch correction, proper count normalization, and generating quality plots. Bash and Linux. Most bioinformatics work happens on Linux systems and compute clusters. Submitting Selurm jobs, writing loops and conditionals, using awk and sed for file processing, and automating entire pipelines. Now here's the secret nobody tells you. Employers would rather hire someone who's really good at Python and understands biology than someone who's mediocre at 5 different languages. Depth beats breadth every time. Pick one type of analysis and learn the complete pipeline. That's worth way more than surface-level knowledge of 10 different things. Depth over breadth. Remember that. Now let's talk about GitHub. This is your actual portfolio. And if your GitHub is empty or messy, you're losing opportunities before you even know about them. Let me show you the difference between a bad GitHub and a good one. Bad GitHub looks like this. Random files scattered everywhere. No README files. Unclear project names like project_final_v2. No documentation explaining what anything does. Everything looks half-finished and abandoned. Recruiters look at this for 10 seconds and move on. Good GitHub looks like this. Clear project organization. Professional README files for every project. Descriptive names that tell you what the project actually does. Full documentation. Everything looks finished and professional. Same person, same coding ability. Completely different impression. Here are 3 projects you should absolutely have on your GitHub. Project 1. A complete data analysis pipeline. RNAseq is probably the easiest to showcase. Download some public data from TCGA or GEO. Process it from raw FASTQ files all the way to results. Create visualizations. Show the whole workflow from start to finish. Project 2. A tool or script that solves a specific problem. This could be as simple as a FASTA file parser or a quality control tool, but make it clean, documented, and reusable. Show that you can build tools other people can actually use. Project 3. An interactive data visualization dashboard. Use R, Shiny, or Python, Plotly, or Dash. Make it interactive. This shows you can communicate results, not just crunch numbers. The key to all of these: make each project look finished. Add a README that explains what it does, how to use it, what you learned. That README file matters more than your actual code quality in a lot of cases, because it shows you can communicate. This is your minimum portfolio. You don't need 20 projects. You need 3 good ones that are fully documented and look professional. Pick one from each category and build them out properly. Quality over quantity. Resume time. Most of you are doing this wrong and it's killing your chances. Let me show you 2 resume bullets. First one says, Analyzed biological data using Python. Boring, generic. Every single applicant says basically this exact thing. Second one says, developed Python pipeline reducing RNA-seq analysis time by 40%, processing over 500 samples from TCGA database. See the difference. Specificity, numbers, impact. Here's the formula that works, and you should literally memorize this. Action verb plus specific skill plus quantifiable result. Not used R for analysis. Instead, performed differential gene expression analysis using DSEQ2, identifying 200 significant biomarkers associated with drug response. Not built a database. Instead, designed PostgreSQL database schema, managing 50,000 patient genomic records with optimized queries reducing lookup time by 60%. Every single bullet point on your resume should follow this formula, and if it doesn't have a number in it, you're probably doing it wrong. Now here's something frustrating. 75% of resumes get rejected before a human even sees them. Why? Because companies use ATS, applicant tracking systems, robots that scan your resume for specific keywords. If the job posting says NGS data analysis and your resume says sequencing work, the robot doesn't recognize that as a match. You could be perfect for the job, but the system rejects you anyway. So you need these keywords in your resume. And I mean the exact keywords from the job posting. Python, R, Bash, SQL, Git. NGS, RNA-seq, Bioconductor, GATK, SAM tools. Machine learning, statistics, Linux. Genomics, proteomics, data analysis. Look at the job description carefully. What specific tools do they mention? Make sure those exact words appear in your resume where appropriate. Don't keyword stuff. That's obvious and dumb. But do make sure you're using the actual terminology that the field uses. LinkedIn is basically your 24/7 job application. Recruiters are searching LinkedIn constantly for candidates, even for positions they haven't posted yet. But here's the problem. Most people's LinkedIn headlines look like this: Bioinformatics graduate seeking opportunities. That's terrible because recruiters don't search for seeking opportunities. They search for specific skills. Your headline should look like this: Bioinformatics scientist, Python, NGS, machine learning, genomics, and drug discovery. See the difference? The second one is full of keywords that recruiters actually search for. If your headline doesn't have the right keywords, you literally don't show up in their searches. You're invisible. Quick LinkedIn checklist: keyword-rich headline. Fill out your skills section with at least 10 relevant skills. Link to your GitHub profile. Join bioinformatics groups and actually engage with content. Connect with people in the field. Recruiters use LinkedIn more than job boards to find candidates. Make yourself findable. Okay, now let's talk about interviews. Technical interviews in bioinformatics are different from pure software engineering. Yes, you might write some code, but they care more about whether you understand the biology and the computation together. Common questions I've actually gotten. Explain how you'd analyze RNA-seq data from start to finish in brief. How would you handle batch effects in your dataset? Walk me through a project where something went wrong and how you fixed it. What tools would you use for variant calling and why? Notice these aren't write a sorting algorithm questions. They're testing whether you can think about biological problems computationally. Here's how to prep. Pick 2 or 3 of your projects and know them inside and out. What was the biological question you were trying to answer? What tools did you use and why those specifically? What were the results? What would you do differently now? Be ready to walk through your thought process, not just recite what you did. And for behavioral questions, use the STAR method. Situation: what was the context and background? Task: what specifically needed to be done? Action: what did you do to address it? Result: what was the outcome? What did you learn? Real example from me. I had a genomic data set with 30% missing values. That's the situation. I needed to clean it for downstream analysis. That's the task. I researched best practices and used multiple imputation with the mice package in R. That's the action. This allowed us to retain 95% of our samples, and we ended up identifying 50 significant genetic variants. That's the result. See how that tells a complete story? That's what they want to hear. Practice this format with your own projects so it feels natural in the interview. All right, so what do you actually do right now after watching this? Here's your 4-week action plan. Week 1 and 2. Pick one project idea from what I showed you and build it. Make it clean. Write a professional README. Push it to GitHub. Don't worry about perfection. Just finish it. Week 2 and 3. Update your resume using that formula I gave you. Action plus skill plus result for every bullet point. Get someone to review it. Seriously, have another person look at it. Week 3 and 4. Optimize your LinkedIn. Fix your headline with keywords. Add your skills. Start connecting with people actually working in bioinformatics. Join relevant groups. Week 4 and beyond. Start applying. But here's the key. Quality over quantity. 10 well-targeted applications with customized resumes beat 50 generic spray and pray applications. Track everything in a spreadsheet. Company, position title, date you applied, which version of your resume you sent, current status. This keeps you organized and helps you see patterns in what's working. And remember, getting your first bioinformatics job is the hardest part. After that first job, it gets way easier. That's part 2. You now have everything you need. You know what skills actually matter. How to build a portfolio recruiters look at. How to make your resume get past the bots. How to optimize LinkedIn. How to prepare for interviews. Between Part 1 and Part 2, you have the complete roadmap. Where to find jobs, what they pay, what skills you need, and how to actually get hired. In Part 1, we covered the bioinformatics job market, salaries, job types, hiring companies, and where to find roles. If you haven't watched it yet, check the link in the description and watch it next. In the next video, we're diving into something big: AI in bioinformatics. The myths, the reality, and how to make yourself more valuable than AI in this field. That one is going to change how you think about your career strategy. Drop any topics you want covered in the comment box. If this video helped you, smash that like button. Subscribe to BI23 for more bioinformatics career content. Thanks for watching and good luck with your job search. You've got this.